But we want to shift gears now to talk about a very interesting way of regulating genes that can be sometimes a little challenging to visualize. This particular method is really found in prokaryotes, a few eukaryote exceptions. It's called an operon. An operon is a fancy way of regulating genes, and it's usually made up of a few genes that can involve enzymes. Remember that enzymes are proteins with the ability to break down or build up the substances that they act on. Now let's talk about some key players in an operon so that we can see some gene regulation here. First, RNA polymerase. It's a builder enzyme actually, because RNA polymerase is an enzyme. In biology, a lot of enzymes end in that ASE. RNA polymerase is needed in order to start transcription. Remember that transcription and translation are steps in protein synthesis. Protein synthesis means to make proteins, enzymes in this case. The thing about RNA polymerase, though, it gets a little confusing for RNA polymerase without somewhere to bind. If you watched our DNA replication video, you learned about DNA polymerase and how it had to have a primer to know where to start. Well, RNA polymerase needs a promoter. A promoter is a sequence of DNA where the RNA polymerase can bind. So you would think, okay, you got your RNA polymerase, you attach it to a promoter, and boom, you make your mRNA, which eventually will be used to make a protein. But there's this other sequence of DNA called an operator. The operator is a part of the DNA where something called a repressor can bind. The repressor, if bound to the operator, blocks RNA polymerase, and poor RNA polymerase cannot move forward and no mRNA can be made, therefore no proteins. So let's take a look at our setup here. This is an example called a lac operon. Notice there's a promoter region of the DNA, the operator region of the DNA, and there are 3 genes here that code for enzymes that help in the process of breaking down lactose. Lactose is a sugar. If lactose sugar is around, bacteria want these enzymes to be made so that they can use them to break down the lactose sugar. Then they can metabolize it. Fed bacteria are happy bacteria. Here's the repressor. There's actually a gene here that codes for producing the repressor. See this gene that we call I? It has its own promoter. Well, this gene codes for the production of the repressor. So why do we need this repressor? Well, it's wasteful to make things that you don't need. If there's no lactose, it wouldn't make sense to start making enzymes that work together to break down lactose. It would be a waste. The enzymes would just sit there. So if lactose is not present, then the repressor binds to the operator, and this blocks RNA polymerase. So mRNA cannot be made, and therefore the proteins, the enzymes in this case, cannot be made. But if lactose is around in the environment, something pretty cool happens. The lactose, remember that's the sugar, binds to the repressor. This changes the repressor's conformation. Try as it might, the repressor cannot bind to the operator. RNA polymerase finds its promoter, binds, and transcribes to make the mRNA from the genes on the operon. That mRNA will be used to make enzymes to break down the lactose sugar. And bacteria like to eat, so that makes them pretty happy. We have to say that we think it's pretty impressive to think about all the gene regulation that occurs in cells. And if you find it fascinating, know that there are careers that focus on gene regulation and understanding how genes can be turned on and turned off. Because by doing that, we can gain a better understanding of a variety of diseases that have gene influences in the human body. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.